Hi, we're going to talk about how to do the mark recapture technique, and we're going to be doing a simulation in my labs. This is Sue Glenn, and so we're going to talk about how we're going to what the technique is, how you collect the data, and then how you analyze the data. We'll be doing a simulation lab where we'll be using toothpicks outside instead of actual animals, but the techniques and the calculations are still the same. So when you're trying to estimate the population size of something, animals are nice compared to plants because sometimes it's hard to tell, is this an individual plant or not? Plants are clonal, so they could be setting out shoots from the roots of trees or trying to figure out how many grasses you have in the lawn. It's difficult to figure out what is one individual plant. Animals, it's pretty clear to see what is an individual penguin, and I can tell them apart. However, mobile animals are also moving around. So did I already count this one? Maybe they all look the same, and I don't know whether I've already got that one. They hide from ecologists, so they don't want to be seen or counted, so you don't know how many you have. Or they might be difficult to catch. They might be up in the tops of trees or down underground, and it is difficult to get population sizes. Coming up with standardized methods for capturing and observing the animals is going to differ with the different type of animal that you're dealing with. But I can usually at least get some or count some. So I might be outside and I can see three or four butterflies, or I can get my net and catch 10 butterflies. I don't know if I've got all the butterflies. Do I have half the population? Do I have 90% of the population? But I can see if in this area I got 10 butterflies and then over in that area there were 25 butterflies, that there's differences in the relative abundances in the different areas, but I still don't know what the absolute population is. So mark recapture techniques allow you to estimate the absolute populations in an area to get an idea of how many the actual population size is. So the methods are pretty straightforward. You go out and you capture as many as you can find, and you're going to mark them. So you're going to identify that I've already seen this guy before. So here's a Fish and Wildlife Service officer who is marking ducks by putting leg bands on them. I thought that was a cute picture. Or you could mark turtles by putting a dot of paint on its shell. Or you could be catching a bear and putting ear tag on the bear. So there's a number of different ways you can mark the animal. Or it could be something very simple and easy, like I could catch a mouse in a mouse trap, a live trap. I bet my live traps with Snickers bars because everybody likes a Snickers bar. And the mouse will, when I get the mouse out, I pick them up and I can just clip toenails on one little paw, and that way if I find that mouse again, I know, oh, it's the one I gave the manicure to. Once you've marked them, you release them. You let them go. So you don't hurt them in any way. You're just going to let them go and as close to this place where you found them so that they're going back into their population. Give them a little bit of time to mix, everybody calm down, and then you go out on a second collecting trip and you get a second sample. So there's a capture, there's a mark and release stage, and then there's a recapture stage. When I go out for my second sample, let's say everything I find has my mark on it, then I'd have a pretty good idea that I had caught all the population and my number of my first sample is pretty close to my population size. If I go out the second time though, and I catch 100 butterflies and I only see one with my mark on it, then I have a very good idea that I didn't get most of the population. There's a lot more out there. And so we make the assumption then when we're estimating the population size that the proportion of your marked ones that you find in your second sample would represent the proportion of marked ones you put back into the original population. So the way this is shown in the equation, that R is how many that had my mark on them, that I recaptured them, and they were in my second sample. And S is the size of my second sample. So if I see a lot of the ones I already had before, this is going to be closer and closer to 1. M is the number of animals I caught in my first sample, so these ones were all marked and then released back into the environment. And they're going to be then a proportion of the total population size. So we know how many are marked, we know how many I caught the second time, and how many from that second catch actually had my mark. So the one thing that we are trying to determine is going to be the actual population size. And so you can solve this for n. So you can determine the actual population size based on those other three numbers. We just rearrange the equation. So if you rearrange the equation, what you're going to be doing, you want to have n at the top, and those guys moved on to the other side. So we're going to move n over here, r there, and s there to rearrange the equation. Here I've rearranged the equation now, so we can see that we can estimate the population size n by simply multiplying the number of marked individuals, that was the size of your first sample, times the size of your second sample, and then you divide that by how many you recaptured with your mark in the second sample. So the smaller the number of ones with your mark in the second sample means the bigger your population estimate is going to be. So the smaller this R is, marked animals recaptured, the larger your population size estimate is going to be. So if you don't see any of them again, or very few of them again, that means that this number is going to be much larger. You could use the Lincoln-Peterson index then to calculate estimated population size. Here's an example. A biologist nets 45 largemouth bass from a pond. So his first sample that he is going to mark is going to have 45 bass in it. He marks them with a fin tag and then releases them back into the pond. Then they catch their second sample, they catch 58 bass. And when they count up how many had their marks on them, they found that 26 of them already had their marks. So you can plug these numbers into this equation. It's going to be 45 times 58 divided by 26 to come up with an estimate of the population size. One of your assumptions then is that you have a closed population, that you don't have any of your marked individuals or any of the population leaving the area. So it's not like I marked a bunch of frogs and then half of them left the site, and that you don't have a bunch of new ones coming in, so that you're assuming with a closed population that we have no emigration, no loss of individuals from your population, and no immigration, no new individuals coming into your population. That's in contrast to an open population where individuals enter and leave between surveys, and you could also have individuals dying and giving birth between the surveys. So an open population would violate the assumptions of this estimate that you're making. So how do you find closed populations? How do you meet this assumption? Well, you might have a closed population where there's a dispersal barrier. So it could be that there is water around the area that, like on an island, that prevents the organisms from coming and going easily, or mountains, some sort of physical barrier. You might have philopatry where the organism has a tendency to come back home, to stay home, so that, salmon will always go back up the same stream that they were born in, that they were spawned in, to go lay their eggs. And also, if you pick a large enough area, then the idea of things coming and going is minimized. So, and that's the most common solution to the assumption of a closed population, is survey a large area, because we don't have an island and we don't have species that show a homing instinct. The other thing that makes a population closed is having a slow reproductive rate and a slow death rate. So you're not having something that only lives for 24 hours or that is producing huge numbers of offspring between your samples. And if we don't have something that has a slow reproductive rate or death rate, we make sure that the time between our surveys is relatively short compared to their reproduction time and their lifespan. So if you put short in time between the surveys, so if I'm going to estimate the mouse population in the park, I could put my surveys a week apart, and I'm not going to have a lot of births and deaths between those weeks. So having large survey areas and short time between the surveys are the most common ways of meeting these assumptions. So when we look at the assumptions of using these techniques, we've talked about that we're assuming there's no significant birth and death rates between births or deaths between the initial sample and the second sample. There's no significant immigration or emigration of individuals in and out of the population. We also assume that the marks aren't lost, so that they haven't lost their ear tags, or if they're a bear, or let's say you had marked the shell of an insect and it has then molted that shell, or you've marked something that has the mark comes off when it rains or something like that. So you make sure that the marks are not going to be lost. You also make sure that you haven't hurt the animals, so that when you let them go, they don't have a higher probability of survival. Like, you don't want to take a turtle and mark its shell with glow-in-the-dark paint, and then have the predators come and eat them. So you want to make sure that the individuals that you marked have the same probability of survival as everybody else in the population. You assume that when you release them back into the population, they're going to mix randomly with the population again. So not all the guys that were marked are going to hang out in the corner and talk about it, but they're going to mix in with everybody else. And that they don't have fear of getting caught the second time. They're not going to be afraid of a trap. And they're not going to be attracted to it. So my Snickers bar is attracting the mice. If it's a good Snickers bar, the mouse is going to come back again, because it said it wasn't so bad the first time. All she did was hold me by the tail and give me a manicure, and I got Snickers for it. So I'm going to come back to the trap over and over again. So you want to make sure you're not attracting them to get recaptured, so you wouldn't have more and more of your marked individuals getting recaptured. On the other hand, some animals are very difficult to catch a second time. I had a student who worked on bears, black bears, in Banff National Park. And the first time he would be able to catch a black bear, he would hang meat from a tree, and he put a little noose on the ground, and the bear would step in the noose while it was trying to get the meat out of the tree, hanging from the tree. It was very weird looking, the meat hanging from the tree. People thought it was like a satanic cult or something. Anyway, he'd be trying to get the meat out of the tree, and the noose they'd grab around the paw, and then my student could get his wife to distract the bear. You got to bring somebody with you to do that job, and it's hard to find somebody to do that job. So his wife would distract the bear, and he'd be on the other side, and he'd shoot the bear in the butt with the tranquilizer gun. And then they could put the ear tag and weigh them and everything. If they needed to try to catch the same bear a second time, they couldn't use the same technique. The bear would never fall for that more than once. They're too smart. So they would have fear of meat hanging from trees, and say, oh, I remember the last time I went for that. That was awful. So the second time he would make like a mixture of fat and berries, and he would put it on the far side of a big log, so the bear would have to step over the log, and he put the noose right there so that he could catch it a second time. But you want to make sure that you're not causing them to avoid your traps. And the other assumption is that your recapture rates are high enough to support an accurate estimate. And that's the most important one. You want to make sure you get enough recaptures that you can make an accurate estimate. If you don't get enough recapture estimates, you're going to overestimate the population. If you get zero, you can't estimate the population at all. So that one is highlighted because that's the most important one. Because of the sample size issue and making sure you have enough recaptures in your second sample, there's a number of techniques that use repeated samples. So instead of just one recapture, you have several recaptures. And the Schnabel index is one of these. And so that you could go back several weeks in a row. And each time you go, you're going to mark more of the unmarked individuals that you capture. So included in this particular equation, we are looking at this number of the total number of previously marked animals in the population. So you might have marked like 50 the first time, and then your second sample, you might have had 10 that didn't have your marks. So you're going to mark 10 more. So then you have 60 marked animals. So you're keeping track of how many total animals you've marked in the population. And so at the time that you're doing that sample, so this is sample I, and so for each sample, it's how many are marked in the population times the number you caught. And so you're going to add that up for all of your sampling trips. And then you're going to also add up how many marked animals you caught in each sampling trip. And that's going to give you a Schnabel estimate of the population size. I think it's easier to understand that equation if we actually work through an example. So here we have somebody caught and marked 100 animals in the first sample. So I've made a little table, and the first sample would have 100 individuals caught, and they're going to be marked. And because it's the first sample, you have nothing that was recaptured. Um, and the population had 0 marked in it. This is your first time out here. So in our animals in our first sample, then we captured 85 animals in the second sample. Of those, 15 had the marks. So then the remaining 70 were unmarked, and those 70 that we will mark. And so, and how many marked ones were in the population that we knew of? We marked 100 and we released them. So there's 100 in that population. And then in the third sample, captured 105 animals. 25 of those were marked. And we're not going to go out a fourth time, so we didn't worry about marking more of those. And how many marked animals were in the population? Well, we'd released all of these guys back, and so 70 of those were new marks, and we already had 100, so now there's 170. We just added those together to get that number. So in order to do this calculation, we have to multiply those two numbers together, the number that were marked in the population times the number caught. So, so I can make an extra column to do that, and I will call it n times c. And so the first time, because it's going to be 0 times 100, that's going to be 0. But then the second time, I'm going to take 85 times 100. So that's going to be 8,500. And then the third time, I'm going to take 105 times 170, and that's going to give me, second, let me do that, 17850. So for the numerator of the equation, I have to sum that over all the samples. So it's going to be 0 plus 8,500 plus 17,850. and that totals up to 2,350. For the denominator, we're adding up how many of the uh, recaptured ones we got at each sample. So that's adding up this 15 and 25, which is going to be 40. So that's the sum of that. That's the sum of that. And in order to calculate the population size, we're going to divide the sum of the marks times the sample size divided by the recaptures. So in order to get that number is going to equal the 26,350 divided by the 40. So the estimate of this population size ends up being 659. So if you're in my class, you have your Ecology lab kit, which includes the lab book, and you're going to need to take that out with you because there's a data sheet and it has all the detailed step-by-step instructions of what you're going to be doing. What you're going to be needing to take out is a marker to mark your toothpicks with. You have a box of toothpicks in your lab kit. And it would be really nice to have a helper who will come out there and help you set up, but also to distribute your toothpicks. Not only initially put them out so you don't know where they all are scattered, but also after you've marked them each time, they can put them out again so that you don't see where they're being scattered, and just don't watch where they put them, and you'll get a more accurate use of this. You're going to find an area. I would do it out in a lawn in a park, grassy area where you'll be able to see your toothpicks. And you can just mark the four corners of the area with your 4 stakes that you have in your kit. And or you can use any kind of sticks that you want to mark your 4 corners of your area, and then have your friend distribute your toothpicks within that area. Also bring your phone with you. You're going to need your timer on your phone, and you're going to need your camera. And don't forget to turn on your weather app notifications in case a storm's coming in. You need the lawn area, and bring some water with you, bring some sunblock. But you're not going to spend much time out. It's not going to take you that long to do this. So your time outside is in the sun is not going to be too bad, but still read the safety guidelines in the lab book before you go out. So the first step, you locate your site, take a picture of it, have your friends distribute your toothpicks without you watching. You can watch some YouTube videos. Oh, I've got some lectures you can watch. That'll be great. And then start your timer. Set it for 4 minutes. So go to your timer on your phone and set a 4-minute timer, or have your friend do it. And then go out and pick up as many toothpicks as you can find in that 4 minutes. So capture as many as you can find. And the only reason we're timing this is so that we have consistent search effort at each sample, which is like you would put out the same number of traps, you'd spend the same amount of time sampling for the resamples as the initial sample. Take the toothpicks, count them, and you're going to record that in the lab book. Then mark them with your marker and take a picture so we can see how many toothpicks you had and what kind of mark you put on it. Because maybe the color mark makes a difference. Maybe some are easier to see than others. And then give them to your friend and have them spread them out again randomly throughout the area. And so when I spread these toothpicks out, just explain it has to sort of cover the whole area. I just walk back and forth and zigzag my way across the area, dropping a few as I go. If it's windy, it makes it easier because they'll fly randomly around. And don't watch while they're doing it. Once the toothpicks have been released back into the wild, then set your timer again for 4 minutes. Go out and pick up any toothpicks you see. So this time you're picking up anything you see again, whether it's one with your mark or one without your mark. Because remember, we're looking at what proportion of the total you pick up this time actually have your mark. So you're going to count the total, and you're going to count the ones with your mark and take a picture of that so that we can see what proportion of it actually had your mark on it. And then the ones that are unmarked, before you re-release them, you're going to have to mark them as well, and because we're going to need to know how many marked ones are out in the population, they all have to be marked. And so you can use the same marker, you can use the same mark. It doesn't matter. You don't have to have a different color or a different kind of mark each time. So give the toothpicks back to your helper, have them spread them out randomly, watch some more of a video, then start the 4-minute timer again and pick up as many toothpicks as you can find. So you're going to capture the marked and the unmarked ones again, just as many as you see. And don't forget when you're done to pick up whatever you put down for the corners. So if you put your stakes out there, go pick up the stakes. And that way you've not going to litter the place except with toothpicks. Luckily for the toothpicks, they are biodegradable. Next time they're mowing the lawn in the park, the mulcher will mulch those all up and you're not really leaving any problems there. You're going to, from this third sample, count the total and count how many of that total have your mark and take a picture of this last sample. So you're going to take a picture of every sample and you're going to put that on your blog. So my students make up blogs for reporting their lab results. And then this is a picture of the page 28 in the lab book. This is your data sheet where you're going to be recording. So you got the date, time of day, whether it's sunny or cloudy or rainy, that can make a difference to how well you see the toothpicks. The date, the time of day, the angle of the sun makes a difference to how well you see the toothpicks. The temperature, probably not a huge difference in this case. And then describe your location and take a picture of it. Then put in your number of toothpicks that you collected in the first round, what mark you put on them, and take a photo. Then your second sample, and then how many had the mark, and how many did you add marks to and photograph it, and the same thing for the third sample. Also write down somewhere how many toothpicks your helper put out, and that is written on the box of toothpicks. So once you've got the data on your lab book data sheet, you're going to send me the data. I'll send you a link for an app that you can input it into our data form. This way I can make your data available to everybody else in the class, and also update your blog with information about your study site and a picture of it, and you can even put in a map of it, and then the pictures that you took of each of your samples, and then describe the site, the date, the time, what kind of color mark you're using, who was helping you, whether there are any other circumstances that people would need to know if they want to use your data. Then you need to do the calculations. So we'll do that the next week. So when you're doing your calculations, you're going to set up the data tables. And you're going to calculate the Peterson index using the data from the first and second round. And then you're going to use all 3 rounds data in order to calculate the Schnabel index. So I've got the equations here for the 2 indices using the exactly the same values for the different variables, symbols for the different variables. This is how I would set up the data set in Excel. So I would have a column for sample 1, 2, and 3. I would have a column here for how many you caught each sample. So C1 is how many you caught in the first sample, C2 is the number in the second sample, C3 is the number in the third sample. Then how many of those caught are recaptured? So 0 obviously in the first sample, but how many had your mark on them? How many did you end up putting marks on? So everything you caught the first time will get new marks, but then this is how many unmarked ones you marked in the second sample. You don't have to worry about marking ones after the third sample because we aren't doing a fourth sample, but if you're doing a fourth sample, don't forget to include that. And then how many marked ones are in the population? So when you did the first sample, nobody had gone out there before you and marked ones, but the number of marked ones in the second sample were how many you marked the first time, and then how many marked ones are in the third sample are how many you marked the first time plus the second time. So it's both these values added together here. Then when you're doing the Lincoln index, we just need this sample size times this sample size divided by the number with your marks in the second sample, and you can calculate that population in estimate. And then the Schnabel index, remember we need to multiply the number previously marked times the sample size at each level. So I just added a column in Excel here where I'm going to take the number marked in the population times the original or the number that you catch. And obviously for the first time that number is going to be 0, but then you'll have values for the second and third time. Then you're going to total that column so that you can get the numerator, and then you're going to total the recapture column to get the denominator. So those will be that one's going to go there, and that's going to go there. So that's how to set up your table in Excel and get your calculations. My students will update their blog with the table that we've just done. So you can see this is from the example we were working on earlier in the presentation. I put in my data table. I've put in my estimates, Lincoln-Peterson and Schnabel. What I want you to do is write a full caption for the table so that it explains enough that's going on in that table that people know what you did and what it means and what all the little C's and R's are. And then I will also ask you to look at other people's numbers on their blogs and come up with some hypotheses that you could test using the class data. And there'll also be an assignment in e-learning where you're going to submit your results and submit the spreadsheet where you do these calculations and answer some questions. Doing a lab like this, you should be able to compare your estimates to the true population size because you know how many toothpicks you started with. And if the values differ, think about why this occurred. You should be able to come up with reasons for that. And then other questions that you should be able to answer. We do a lab exam at the end of the semester. You should be able to answer any of these questions about the mark recapture technique. How do your estimates compare to the true population? What kind of animals would this work well for? What wouldn't it work well for? Like why? Did the Schnabel index give you a better estimate of the actual population size than the Lincoln-Peterson index? And if so or if not, why? How could you increase the accuracy? Can you think of better ways to estimate the numbers of animals that are moving around in a population? So we had a closed population. We met the assumptions of these techniques, and we still probably have some inaccuracies. So accuracy is how close your actual value is to the truth. And since you know the truth, you'll be able to figure out accuracy. The other thing to think about is precision. And precision is how different are the estimates from different students doing exactly the same thing. Are they all kind of getting similar answers, or is it very imprecise, and do we get a lot of variability in the data? So you can think about those as well. So thank you. Sorry we couldn't sample something cool like wolves, but I think this gives you a hands-on experience. You get to spend a little time outside and you get to do these calculations. Mark recapture is widely used for estimating populations along with the indexes you've learned here. A lot of other indexes have been generated, and I think if you're reviewing any papers dealing with population sizes, you're going to be running into discussions about these techniques. So hopefully you get a lot out of doing this exercise.